after successful completion of 11 programs, we have decided to invite another expert to be here with you all today, a program which we organize once in every 2 weeks, every Wednesday, once in every 2 weeks, Wednesdays at 7 p.m. The expert that we have invited today will be introducing to you in a while. The overall idea about this webinar series is to make sure the general public, scientists, academia, students, and all, everyone in Sri Lanka especially, to clear doubts about the issues that they have with respect to certain subjects of national importance, and also to make sure we upgrade, in other words, our knowledge with respect to current scenarios and looking at the future. That's the overall objective. And without much ado, let me now invite the president of the Alumni Association of Faculty of Agriculture at University of Peradeniya, the person who planted the seed, brought in the idea to make sure this public webinar series is conducted for people to clear their doubts, as I told you a little while ago, on very important subject areas of interest. Dr. Tushara Vikramarachchi, over to you, sir, to welcome the participants formally.
Thank you, sir. Thank you so much. Today actually very important day for us because this is a 12th consecutive public seminar. Today we are going to conduct the 12th one. That is the monthly ones, then almost 12 months. Now actually today we because we are inspired because of our participants. Every day it is nearly 50. Today we can see it is more than 50 because that is today's very special scientist, none other than that is the Professor Warshi Dandeniya. No introduction is required actually because everybody knows her because of instrumental in very challenging decision in the Sri Lanka. She's the one of the several the academias who came forward to make the against the certain challenging decision of the country. But we pay we convey our regards to the Professor Warshi as well as all the participants. Actually, everybody with us in every month our public webinar. Then we are encouraging ourselves as Faculty Alumni Association, Faculty of Agriculture, to bring you many more things to to enhance the knowledge in agriculture. Thank you. Today also very exciting the webinar from the Professor Warshi, and thank you Buddhi Marabesa because she's the always for this 12 webinar she the volunteered to do the facilitating of this seminar series. Then thank you. Then over to you, sir.
Thank you very much, Dr. Vikramarachchi. Of course, it's a pleasure to be here and moderate these sessions as well. Subjects of extreme importance in the national context. Without much ado now, let me brief you about the mode of operation of this public webinar series. Please do keep in mind that every time when we had a webinar like this, of this nature, it's a planned program. Planned in the sense the moderator and the speaker discusses, once we reach an agreement about the topic, we discuss about the key burning issues, and the presenter will make the presentation based on questions, pre-planned questions agreed upon by the moderator and the speaker. It's just to make sure there will be a flow. And also, I must tell you that we have crafted the questions based on the needs of the society after discussing matters a lot, and by understanding the sort of issues that prevail among the general public, especially on this particular subject. So it's basically a kind of QA session that goes on in terms of presentation. As for the audience, the participants can ask questions always, but will not be given a chance to ask a question verbally. If you have questions, please post it on the chat box. So at the end of the presentation, as the moderator, I will take the responsibility to post those questions to the speaker today. So that will be the mode of operation. I kindly request you to switch off your mics, or in other words, mute your microphones, and let's communicate through the chat box without interfering the presentation that's going to have in a while. Well, our speaker today, as Dr. Vikramarke said, need no introduction. So I'm not going to give you lengthy introductions about the speaker. Is none other than Professor Warshi Dandeniya, who is at the present context is not in the country. I must thank in that sense. So Warshi, you can definitely let the people know as well where you are right now. And she is a professor in soil science, and especially at the Department of Soil Science of the Faculty of Agriculture, University of Peradeniya. And topic that she has chosen today, facts and myths about biofertilizer in agriculture. Let's rely on facts and bust the myths. That's the most important part. So, Professor Dandeniya, let me take the liberty in asking you the first question. I'm sure you can then start sharing the presentation and move ahead. Simple question at the very beginning. What is it all about? What is this biofertilizer? I think that will be a good start so that everybody in this audience will be in a common platform to go ahead. Over to you, Warshi.
Thank you, sir. I hope you all can hear me clearly.
Loud and clear.
Yeah. Let me start sharing my screen. Can you see it in the slide share mode, sir?
Yes, we see it clearly, Warshi. Go ahead.
Okay. Yeah. And first of all, I must thank the Alumni Association for inviting me to deliver a presentation in this very important webinar series. And as Professor Buddhimarame mentioned, I'm not currently in the country. I'm taking my sabbatical leave in Hong Kong. I did not leave the country, like not ran away. Some people think like that. So I must clarify, I am currently in my sabbatical leave working in Education University of Hong Kong. So thank you very much for giving this opportunity to talk about this very important topic. So as Professor Marambe asked, what is biofertilizer? Let me tell you what is biofertilizer and bit about its history so that we can all understand what are we talking about today. So if you look at the definitions, there are many definitions about biofertilizer. I have taken one very commonly used definition in scientific literature. That is, you can see that I have highlighted a couple of words in this definition. So it's a material that is that contains living microorganisms that can be applied to seeds, plant surfaces, or soil, or can be used to colonize the rhizosphere, that means the root zone of a crop. And by doing that, these live organisms will increase the supply or availability of primary nutrients to the crop. So this is the basic definition we have, a scientific definition about biofertilizer. And let me share you another definition according to our country's context, because we are in the process of developing the standards for biofertilizer specifications. So in that draft standard, I think it is at the finalizing stage right now. According to that draft standard, again, the definition says it's a single or a consortium of beneficial microorganisms. And then there are some other important functions that we are expecting from these organisms mentioned, but ultimately increasing the plant access to nutrients. So when we say a fertilizer is applied to increase the nutrient availability for a plant. So a biofertilizer is a substance that contains living microorganisms that will be used to increase plants' access to nutrients. So this is the basic definition of a biofertilizer. And what is it made out of? Of course, it contains living organisms. Usually the candidates we consider in developing biofertilizers are the nitrogen fixers, phosphate solubilizers, Then potassium solubilizers, often referred to as silicate bacteria. And then presently, there are some countries using micronutrient mobilizers, especially targeting to mobilize zinc, molybdenum-like micronutrients. And in addition to providing these nutrients, there are some other benefits of these bacteria and the organisms we use in producing the biofertilizers. And those benefits are some of these candidates we consider for biofertilizer development may contain plant growth promotional characters, like producing substances similar to plant hormones, increasing the root development or shoot growth, etc. So if we look at the taxonomic groups that we all know, these candidates belong to different groups of bacteria, fungi, and microalgae. So these are the organisms that we consider in developing biofertilizers. And let me brief you a bit about some misconceptions related to biofertilizers. Because if you go to market, there are sometimes, even though in Sri Lanka we are still not we have only one product that has been approved by the government authorities, that is a biofilm biofertilizer. But if you go to market, you can still find some unauthorized products referred to as biofertilizers. And some of these material, if you take a look at the composition, it only has either the microorganisms who can promote plant growth, like those who produce auxin or cytokinin or gibberellic acid, like plant hormone kind substances, but not necessarily providing nutrients, like there's no nitrogen fixers or P solubilizers. Therefore, we call such products as biostimulants, that is stimulating plant growth, but not necessarily a biofertilizer. And also, there are some other products which contain the metabolites, that mean the enzymes produced by bacteria and fungi, separated or extracted and concentrated and sold in the market as growth hormones. So those contain the metabolic products of organisms, no live organisms. So those are also biostimulants, but not biofertilizers. And also there are some EM mixtures and specific microbial products that we target to apply when we are doing composting or straw decomposition like activities. Those are also not biofertilizers. Those are decomposers that are there to increase the microbial decomposition activity. Okay, so these products are not biofertilizers. I think it's good to have this clear distinction, what is a biofertilizer and what is not a biofertilizer. So in summary, all the biostimulants are not biofertilizers, but biofertilizers can have biostimulant properties. So let me stop answering this question, giving you a brief history about biofertilizers as well. So if you look at the history of biofertilizers, it doesn't have a long history like chemical fertilizers. So there have been attempts to isolate organisms and apply those to plants to improve plant growth since 18th century. Of course, when we came to know about microorganisms, John Baptist and the scientists, leading scientists, they tried to isolate nitrogen fixers and use it. However, we can see that the use of these bacteria and fungi in targeting to increase the nutrient availability for a crop actually happened in early 1950s, starting in Soviet Union and in China. In their farms, they have started using these nitrogen fixing bacteria and phosphorus bacteria and potassium solubilizers to improve crop production. And also in Africa, in South Africa, and in Zambia, we can see the introduction of the microbial inoculum nitrosam in 1992, but they started like releasing of legume species which are very responsive to native nitrogen fixers in 1963. So we can see like it's not a very long history. We don't have 100 years of history for biofertilizer, but at present in 24 countries around the world, biofertilizers are being produced in commercial scale. I think in China alone, they have about 650 different products of biofertilizers. Okay, so this is a brief introduction about biofertilizers.
Thank you very much, Professor Warshi Dandeniya. One important message that we got out of our presentation: all microbial products are not biofertilizers, though they have been used in agriculture for various reasons. Well, Warshi, we are talking about chemical fertilizers, organic fertilizers, biofertilizers. I mean, a lot of terminologies come up with respect to agriculture, especially when we speak of commercial level or non-commercial level. I mean, everything has a commercial value, as we know that. Before going into looking at, or to have a deep dive in biofertilizer, I think it's good at this particular moment for you to let us know what is the real use of fertilizer. I'm not referring to biofertilizer, Warshi, here. What is the real use of fertilizer? So in other words, why we use fertilizer in commercial agriculture? Because I heard many people telling that, well, Singaraja forest, it grows without having any fertilizer being added. Interesting statements. Over to you.
Yeah, thank you for this very important question, sir. So I think it's very important for us to understand what is an agricultural system and what is the main character. How is it different from a natural forest system? Okay, so if we take a look at our agricultural systems, what we do is we grow crops and we remove some harvest. Okay, we remove part of the biomass as harvest. So I want to emphasize one word that you mentioned, that is commercial agriculture. Because agriculture can be done in different level, like subsistence level, like even home gardening activities is a different type of agriculture. But if we are cultivating crops, especially as like thinking of earning money in a commercial scale, that means we are usually utilizing the available resources in a very intensive manner. So in that sense, we are frequently removing large quantities of biomass as the harvest. So when we are removing a biomass as harvest, one thing we need to remember is we are removing nutrients from the system. Therefore, this agricultural system, the cropping systems, especially at commercial scale, we can consider it as an open system. It's not closed like in a forest. So there's not enough time to regenerate the soil and supply nutrients in the natural methods to the soil in a commercial agricultural system. So because we are removing nutrients, of course, there's a need of supplying nutrients to the soil. So let me take you through a few examples to get an idea in quantitative terms. So if we look at the composition in plants, we know there are 16 plus essential nutrients, and out of these, except for carbon, hydrogen, and oxygen, NPK are the next three most important nutrients. So if you look at the composition in plant by dry weight, we consider these NPK, the three nutrients, we call them mineral nutrients because they are taken up by the plants from soil, okay, so, or the growth medium through the root system. Therefore, these three nutrients are required by plants in large quantities, and these three become very important when we consider about nutrient supply to crops. Because when we are removing harvest, when the plant are taking up nutrients from the soil, these NPK are the three nutrients that are going to get frequently removed from the cropping system. Let me give you some quantitative values here. If you take rice, corn, potato, like these different crops, with the harvest, that is the grains that we are removing with rice crop and the corn cobs with the maize cultivation, you can see the amounts of nitrogen, phosphorus, and potassium being removed from the cropping system. So that is when we are expecting the targeted yield mentioned within parentheses. So if we are getting this yield, that means we are removing the mentioned amount of nutrients from the system. So if you think about like, okay, we are not going to supply any nutrients, we are not going to add any nutrients extra, let the system replenish itself. That means naturally the microorganisms, nitrogen fixers should fix nitrogen, then phosphorus solubilizers, potassium solubilizers like other organisms should mobilize the nutrients in the soil. They take time and the quantities they would supply are not a match to the amount that we are removing with these intensive cultivations. Therefore, we have to supply the nutrients in commercial agriculture either as organic or inorganic form. It doesn't matter whether we are supplying it through organic fertilizer, inorganic fertilizer. The bottom line is we have to apply nutrients to the crop field because it is not a closed system. So that is why we have to supply fertilizers, or we have to use fertilizers in commercial scale agriculture.
Thank you very much, Warshi. We cannot starve the crop and expect higher yields from it. Natural, and that we all have to understand. Well, that's a story, general story about fertilizer. Let's come back to biofertilizers now, which is the topic of the day. Now, fertilizers, I mean, it provides nutrients, supply nutrients for plant growth and development. Now, let us know, how is the nutrient supply from biofertilizers differ from other types of fertilizers? I think that will be a question that we have seen coming up from many sources. Warshi, for every forum that we go in, we would like to know an answer from you for that. Over to you.
Okay. Thank you, sir, for the question. So the question is about how these different types of fertilizer I mentioned are different. What is the difference in nutrient supply in these three fertilizers? Let me first brief you about how the nutrients present in the soil. So in soil, we have nutrients in the soil solution, and which is the readily available forms of nutrients. Some of you may know about ammonium ion, nitrate ions, orthophosphate ions, like ionic forms of the nutrients are present, dissolved in the soil solution, and that is where the plants would take up the nutrients. And there are some amount of nutrients in easily mobilizable forms, but they are not readily available for plant uptake. For example, proteins and other carbohydrates, lipids, then calcium phosphates, like precipitated forms, or the nutrients adsorbed onto cation exchangeable sites. These are not readily available for plant uptake. However, through mineralization and like another dissolution, like transformations, those nutrients can come into the soil solution and plant can take up. At the same time, there's a big reserve of stabilized forms of nutrients like humic substances, then primary minerals. Some of you may remember about the apatite, hematite, mineral forms, the feldspar that we talk about. These are the stabilized nutrients. They are not releasing nutrients easily. It will take maybe decades to centuries to release the nutrients in these stabilized forms into soil solution. So in soil, we have these different types of different pools of nutrients. So what happens when we are applying fertilizers? So when we use organic fertilizers, even though compost is not an organic fertilizer, in soil science, we like to refer to it as an organic amendment. But in Sri Lanka and in many other places, it is commonly referred to as an organic fertilizer. So let me take you through what happens when we apply compost or animal manures like cattle manure to soil. So most of the nutrients in the organic fertilizers will enter into the easily mobilizable forms, and part of it will go to soil solution. So the nutrients added through organic fertilizers therefore remain in soil for considerable amount of time because most of it enters into this easily mobilizable pool. So when we apply chemical fertilizers, the common chemical fertilizers like urea, TSP, and MOP. I'm not talking about the 4th generation or the very improved versions. I'm talking about the 3rd generation fertilizers and urea, MOP, TSP, like ones that we are very familiar with. So these fertilizers mostly release the nutrients into the soil solution because they are readily soluble in water. So their contribution to the soil solution is high. And after it enters the soil solution, it will slowly immobilize into these easily mobilizable forms. So this is what happens with organic and chemical fertilizers. So what happens with the biofertilizers? Where does it fit? So if we take a look at biofertilizers, I would like to consider biofertilizers under 2 categories. Those are the nitrogen biofertilizers and P and K biofertilizers. So if we look at nitrogen biofertilizers comprise of nitrogen fixing microorganisms who will bring atmospheric nitrogen into soil solution as ammonium and nitrate. Or the ammonium form, which will convert in the soil into nitrate. So the nitrogen fixers will bring the nitrogen from the atmosphere. And the P and K biofertilizers, that means the phosphorus and potassium solubilizer based biofertilizers, how they would act is these organisms will solubilize the easily mobilizable nutrient forms and release the particular nutrient into soil solution. So what their role is to increase the rate of transformation inside the soil. So what's important to note that here is for them to be active and release enough amount of nutrients to the soil solution, there should be a good reserve of nutrient present in the soil. So when we are applying biofertilizers, we are usually improving the microbial ecology in the soil. And with that, the nutrient cycling get improved and the plant growth get promoted. And as a result, when we are removing the biomass, the nutrient removal also increases. Okay. So with that, what's important to remember is it increases the chance of nutrient mining with time. Nutrient mining means removing nutrients from soil. For example, if you take P and K biofertilizers are converting the easily mobilizable forms into what is there in the soil solution. And as a result, gradually with time, the size of the easily mobilizable nutrient pool in the soil get depleted. Therefore, when we are using these microorganisms as biofertilizers, we should make sure that we have enough phosphorus and potassium nutrient reserves in the soil. Otherwise, we will do nutrient mining. So this is one of our, I would like to share some experimental results to you. So I am talking about these nutrient mining and changes in the nutrient pools with research experience. So this is an experiment we conducted with rice soils. What you see as marked with these red color circles are the 2 treatments where we applied phosphate solubilizing bacteria to soil, and the ADW and CS. These 2 terms refer to 2 different water management systems. And under both water management systems, what we observed was when we apply the phosphate solubilizing bacteria, the first few bars, the stack bars, indicate the readily available and moderately available phosphorus forms in the soil. So the readily available and moderately available phosphorus forms increased in the soil when we apply the phosphate solubilizing bacteria because they are increasing the mobility of phosphorus. Okay, so and these different organisms have different capabilities in mobilizing different complex forms of phosphorus as well. This is, I'm sharing another experience we have we isolated 11 different bacterial isolates from paddy fields, and we observed they have different capacities when it comes to solubilizing calcium phosphate, aluminum phosphate, and iron phosphate, different complex forms of phosphorus. So depending on what form is there in your soil in most abundant, how fast these nutrients get mobilized, and also what sort of bacteria or the fungi you have used in the biofertilizer, how fast the nutrient get mobilized would be different.
Thank you very, very much. That's a very clear explanation. Warshi, when I listen to your presentation, I mean, every time when we use a fertilizer, we anticipate a good outcome of it. And of course, we are always concerned about the performance of the fertilizers. We call it spatially and temporally. Now, what do you think? What will be the performance of a biofertilizer? Or is it consistent across space and time as chemical fertilizers? What is your view on this?
Yeah, thank you, sir, for the question. I think this is a very important question, and one reason, knowing about this concept is very important to understand why biofertilizer has not become popular as chemical fertilizers among farmers. So the reason you will understand when I go through the next couple of slides. So first thing we need to understand, soils are different from place to place. Maybe like as soil scientists, we always like to look at soil, look into soil. But even those who are not familiar with the soil science discipline, you may agree with me that soils are different from place to place. So when the soil is different from one place to another, along with that, the soil properties, chemical, physical, and biological properties, these also changes. And not only that, the microclimate close to the soil environment, it also changed spatially and also temporally. And with all these, the microbial population in the soil would be different from place to place. Let me take you through a few examples using photos, because with photos, it's easy to understand. I have taken these examples from rice cultivated soils in Sri Lanka from the dry and intermediate zones. These photos are from the Soils of Sri Lanka book edited by Professor R.B. Mapa in 2020. And according to these photos, I think it's even though we are talking about rice cultivation, a single crop, we are not now comparing about rice with upland crops. Let's take a look at rice cultivated soils in Sri Lanka. Rice cultivated soils in Sri Lanka, from place to place, there are many differences. You can see from the color, the depth, the horizons that we can see in profiles, there are very many differences. Okay, so in the next slide also we can see, because we cultivate rice in grumu soils, then in reddish brown earth soils, and then there's low humic clay soils, non-calcic brown. There are different associations of these grade groups when it comes to rice cultivated soils. So with this visible variability, you can get an idea, okay, there should be some differences in properties as well. So when we look at these soils, we can say soils are different from place to place, and along with that, properties also differ. And when the soil properties change, it affects the availability of resources for microorganisms and also biotic interactions. Okay, the biotic interactions would also therefore change from place to place. So when we are applying a biofertilizer, which is a mixture of living organisms to soil, whether they can survive and perform in these different places will be determined by very many factors. So the biofertilizer that apply in Gunapala's paddy field may work well, but in Siripala's paddy field, it may not work. Okay, so simple as that. So that's why we can see a lot of inconsistencies in performance of biofertilizers from place to place. So let me share some of our research experience. This is like a biofertilizer we have developed in our lab with phosphate solubilizing bacteria. And this phosphate solubilizing bacteria based biofertilizer, when we trialed it with different farm fields in the dry and intermediate zone in the country, we could see the performance was higher in some fields compared to the TSP application, whereas in other fields the performance was equal or little less. Okay, so it is difficult to get a consistent performance throughout. That is because of the nature of the material itself. It consists of live organisms who are responding to the environment that we are releasing them. Okay, so here's another example. This is in Arlaganville and Dehiatakandia in Mahavali authority farms that we have used these biofertilizers. And in these farms, they have used the biofertilizer together with different rice varieties and different cultivation methods. And two different types of biofertilizers we have tested. One is a phosphate solubilizer based biofertilizer, and the other one is a cyanobacteria based nitrogen fixing biofertilizer, which was developed by Dr. Vajira Balasurya from Wayambhu University of Sri Lanka. So when we look at these biofertilizers, again, we can see inconsistencies in performance. In some places, we can get very good relative yield compared to the DOA recommendation, whereas in other places, the performance is not very good. So these differences are there with time, like in terms of space as well as with seasons. So when we summarize our experience related to the biofertilizer research, we can see with paddy crop, in certain seasons we got very good results, but in some other seasons, the performance was similar to the TSP 100% application, but we were satisfied with the results because we got statistically similar results to TSP application with a reduced rate of TSP application when we supplied the biofertilizer. Okay, so we were able to save or reduce the application rate of TSP in these fields. However, like considering the different biofertilizer research conducted in Sri Lanka, we can see these inconsistencies in results have been very often reported. And this is an experience with chili that we did with in a pot experiment level. Here I am reporting. What you see as yellow color bars in these charts are the treatments that receive the microbial inoculum, which contain phosphate solubilizing bacteria. And just by looking at a glance, what you can see is in the alphisol and the altisol, the two soils, the performance of these are different. Because one reason is, if you look at the table here, the both soils have very high total phosphorus reserves, but in alphisol it was around 800, while in altisol, the RYP, red yellow podocylic soil that we can find in Nuvarelia region, the total phosphorus content was around 5,000, a very high total P content. And the available phosphorus content was also very high there, 172. So basically, no phosphorus might be needed to apply by looking at the available P content. And we could observe that when you look at the response of the crop to the fertilizer applied, which is indicated by the second bar related to NK, treatment NK. So in the alphisol soil, which has low available phosphorus, only 4 ppm, 4 milligram per kilogram, you can see crop response to the added fertilizer. But in the altisol soil, there's no crop response to the added fertilizer. So the biofertilizer also worked well, performed well in the soil where phosphorus response was seen, the fertilizer response was seen. In the other soil, fertilizer response was not very clear. Therefore, the effectiveness of biofertilizer is difficult to comment on. The same thing we can consider because biofertilizers have live organisms. Therefore, these organisms, once we release in a targeted environment, they are subjected to competitors, subjected to graze by predators, So they have to live. Okay, so if they live and perform only, we can see the biofertilizer is effective. And especially if we are using a biofertilizer which contains fungi, we need to make sure there's enough organic carbon in the soil. That means this is highlighting one important fact, that is, we need to know the ecology of the organisms which are in our product. Because if it is a fungi-based biofertilizer, fungi need organic carbon to respirate, produce energy, because it's its main substrate for energy generation. So if you apply fungi to a soil which does not have enough soluble carbon, then it will not be successful. Okay, so if you are applying a fungi-based biofertilizer to a soil, we need to supply support by providing some organic substrates. And when we are using P and K biofertilizers, as I mentioned earlier, the quality and the quantity of phosphorus and potassium reserves in the soil matter. So if your biofertilizer contains calcium phosphate solubilizers, but when you are applying it in a soil in newer area, like place where there may be more aluminum phosphate and iron phosphate, that biofertilizer might not work. So you need to have a good idea about the ecology, the physiological requirement of the organisms, and the characters of the destination where you plan to apply these organisms. So that is why when we look at biofertilizer research all over the world, many researchers have indicated that they can replace chemical fertilizer requirement by 20 to 50%, depending on the targeted nutrient and the cropping system, without affecting the yield or without compromising the yield. So this is the common observation. So if someone claims you can apply the biofertilizer and replace chemical fertilizers by 100%, it is very unlikely. In scientific terms, it's very unlikely. Either you have to supply organic forms of nutrients or inorganic forms of nutrients to support the system. Okay, otherwise it's not easy thing to do. And I would like to highlight a few special characters about nitrogen-based biofertilizers, because unlike the phosphorus and potassium solubilizers-based biofertilizers, nitrogen fixers containing biofertilizers are special in few sense, because they get affected by environmental conditions very much, especially the nitrogen availability and the pH and carbon availability in the soil. Okay, these things matter a lot for nitrogen fixers. The main reason is, I think you all know about production of nitrogen fertilizers. Even we use the Haber-Bosch method, which where we break down the triple bond of nitrogen using very high temperatures and pressure. So that itself indicates there should be very high energy requirement in breaking down the nitrogen molecule to form ammonium. Okay, so nitrogen fixation, even in biological sense, it's very energy demanding process. So if there's available nitrogen in the soil solution, microorganisms will not bother about spending energy fixing nitrogen. Okay, so if we apply nitrogen fixers along with urea, the chance of nitrogen fixers working in the system is very minimal, because there's enough readily available nitrogen in the system. Okay, so in addition, when we talk about symbiotic nitrogen fixers, in addition to the growth requirement of the nitrogen fixers, the requirement of the host plant, for example, the legume or the azolla, the growth requirements of these also matters when it comes to success of that biofertilizer. So in paddy fields, why azolla and abina combination is not very much popular is one reason is in paddy cultivation, after like maximum tillering, the canopy cover is too much. There's not enough sunlight penetrating to the standing water for the azolla to continue photosynthesis. And in that sense, azolla is unable to provide the nutrients to the other symbiotic organisms. Okay, so similarly in legumes also, if the field has molybdenum or nickel-like deficiencies, the plant performance is going to be poor and the nitrogen fixation is going to be poor. So all in all, if we look at this nitrogen fixation and how much of nitrogen can be fixed by microorganisms and provide to cropping systems, by looking at this table, you can get some idea. I just want to highlight, just look at the variability. If you consider the rice, blue-green algae, the variability of the amount of nitrogen fixed vary from 10 to 80. And if you look at the glycine max, the variability indicated as 0 to 237 kilograms of nitrogen per hectare. Okay, so there's a huge variability. So in one field, the nitrogen fixers might be providing enough nitrogen. In another field, it may not. The reason is this variability in growth characteristics among organisms and in their destinations. The requirements, whether the requirements match with the conditions in the destination. Based on that, their performance would be different, and that will result in spatial and temporal variability in the performance of biofertilizers, which is not seen in chemical fertilizers. Whether you apply urea in regosol soil, the sandy soil in Kalpitia, or in a clay soil in Nuerelia, it doesn't matter whether it's Kalpitia or the Nuerelia areas, soil, the urea will release nitrogen in the same way. But it's not so with the biofertilizers.
Thank you very much, Warshi, bringing in a lot of information, a lot of research findings from your own work, as well as elsewhere that we have found in the published form, to clear our minds with respect to spatial and temporal variability with respect to use of biofertilizer. There's one question that came to my mind when I was listening to you, of course, the slides that you presented in the first segment. Well, microorganisms are used for biofertilizer production, that's true. Now, do they often have plant growth promotional traits as well? Over to you, Warshi.
Yeah, thank you for the question, sir. Yes, I mentioned at the very beginning, although the biofertilizers are produced with candidate organisms that target to provide a primary nutrient, some of these organisms have other beneficial growth promotional characters. So let me share some experience again from our research. So what you can see here is the results we obtain based on the phosphate solubilizing based biofertilizer. When we apply to soil, we observe that the biofertilizer it improved the plant growth. And when we applied it to the seeds, we could observe the seed germination, the germination rate, and the properties of the seedlings also getting improved. Okay, so here in this table, I'm summarizing some of the growth promotional characters. So some of the isolates we have used in producing the biofertilizer were producing inidol acetic acid-like substances, like similar to auxin, and some are having protease activity. And in literature, there are research indicating like some of these microorganisms who are used in biofertilizer production produce cytokinin or similar substances, which are plant growth regulators. Okay, and when we look at the germination in the next table, you can see with different rice varieties, we tried the germination percentage and looked at how the germination is improved when we inoculated the seeds. And all the letters highlighted in red color, you can see the numbers. The germination percentage has increased with the inoculation of the seeds. So the biofertilizers, the organisms, the candidate organisms we used in the biofertilizer production very often have these growth promotional characters. And this can be really, this can come really handy in our cropping systems, because let me share this data with you. This is based on our trials conducted with vegetable cultivating farmers. In the farm fields, we had 3 treatments applied, which is, again, this is related to the phosphate solubilizing bacteria-based biofertilizer. We have developed targeting crop fields, vegetable cultivation. The leader in this project is Ms. Kumudu Kumari Navaratna from Horticulture Research Development Institute of the Department of Agriculture, and I'm a collaborative scientist in that project. So in our research, we saw that in certain fields we have a low phosphorus availability. In certain other fields, we have very high phosphorus availability under farm field condition. And so where we have the low phosphorus availability, we see that the crop is responsive to the added fertilizer, as in the first graph. But in the second graph, you can see the values are less than 1. The relative yield is less than 1. That indicate the fields are not responsive to the added phosphorus fertilizer when we add as triple superphosphate in 100% of the recommended rate. But when we apply the phosphate solubilizing bacteria, where we did not see a phosphorus response because the soil already have a lot of phosphorus. But when we apply the phosphate solubilizing bacteria with a reduced rate of TSP, we could observe the plant growth has improved. The yield has improved. And that may be because of these growth promotional activities. So because of the improvement in plant growth, the plant vigor, its ability to tolerate stress, these conditions also get improved, and these additional benefits also contribute to the increased yield in these farm fields.
Yeah, thank you very much. I think that's very, very clear about your presentation and the explanation given with respect to the question that I asked. Well, Warshi, we have now been talking about all good characteristics of biofertilizer, its impacts, how it provides nitrogen, how it provides nutrition, and affects agriculture in a very beneficial manner. But I'm sure everyone would like to know, with such a product, how is that it is being developed, isn't it? So let me put forward a question. Let us know, tell us, how is a biofertilizer developed? Over to you.
Yeah. Yes, it's a very important question, how a biofertilizer is developed, especially when we consider the past 2-year situation in the country. Many products came to the market claiming by different groups that they have developed this fertilizer, that fertilizer, etc. So let's take a look at how a biofertilizer is developed. So development of a biofertilizer has several steps. It's not something that we do overnight. Okay, it's not something that we do in the laboratory, putting few chemicals together and produce like a chemical fertilizer. No, it has several steps. It starts with isolating the microorganisms that are having some targeted functions from the soil or plants. Okay, so for example, if we are targeting nitrogen fixing ability, we have to isolate nitrogen fixers. If we want to take organisms who would colonize the root system inside the root, then we have to sample the plant roots, and collect them. If we are targeting to apply them to the soil, then we need to collect soil samples and isolate. So that's where it starts. So we have to isolate the organisms, and then we do many laboratory screening, characterizing test to see, for example, if you take nitrogen fixers, is it really fixing nitrogen? What are the conditions there should be there for it to fix nitrogen? Whether it requires some nitrogen availability in the system, or what should be the pH, what should be the temperature, and what is the rate at which it is fixing nitrogen. Likewise, we do a lot of laboratory screening, characterizing, and then identifying the isolates. We need to identify the organism up to strain level. I think you might be clear by the term strain level. That means the genus, species, and the strain. Okay, and then it's not only that, but we need to make sure that this organism is safe to handle. Okay, it's not producing any toxic substances or does not create any health hazards to the human beings and the animals. It's very important. There can be a very good phosphorus solubilizer, but it can be a disease-causing organism. In that case, we cannot use it. So therefore, laboratory screening, characterizing, identifying, and testing the toxicology and safety are needed. And then we need to prepare inoculum and test the performance under laboratory and plant house conditions first. we do not directly go for field application because these are organisms we are talking about. And if the organism is an invasive type or have some problems, if we directly go and do a field application, we cannot later go and collect them. Okay, like it's like cut out of the bag. After you spill it, you cannot collect it. So we don't do field experiment until we are sure about the safety of its use and its performance under laboratory and plant house conditions. So after we confirm this performance under laboratory and plant house conditions, then we do field scale experiments and identify the best performing environmental conditions and also the process of inoculation. Should we apply this as a seed inoculant, or should we apply it at the time of fertilizer application at the basal dressing or as a top dressing, etc. So you need to do study about what is the process of inoculation, and then with that we once you come to that step, we have a successful inoculant. And an inoculant is not a biofertilizer. To make it a biofertilizer, we need to consider how are we going to keep this inoculum in long-term storage with some carrier material and formulate it into a fertilizer. So the biofertilizer contain this inoculum plus carrier material, because the carrier material will support the organisms to be alive during the storage and transportation. So then we need to trial on what is the best formulation. Is it a liquid formulation or a solid formulation for this particular inoculum that we develop? And we need to do a series of tests on the shelf life, storage characters, and how the quality changes with the storage for this biofertilizer formulation. And finally, after we prepare this the inoculum into a biofertilizer, this biofertilizer need to be tested again under field condition. So you can see there's a series of steps, not just stop at producing an inoculum, but then converting it into a formulation, a fertilizer formulation, and testing it under field condition. And let me give you a few important facts about isolation of organisms. At the step of isolating organisms, we need to have a clear idea where are we going to apply these organisms. Let me give an example from our humankind. It's very easy to understand this. Now, if you take a person from Sri Lanka, a tropical country, and take that person to Alaska, a very temperate environment covered with snow. Okay, you take directly a person from Sri Lanka and put that person in Alaska or in Iceland and ask the person to work outside, plow the soil. Do you think that person would survive? That person would perform well? Of course not, because the ecological adaptations we have are to survive very well in a tropical environment. So similarly, when we think about microorganisms, like if you want to develop a biofertilizer targeting paddy cultivation in Sri Lanka, you need to collect organisms from paddy fields. If you collect organisms from Singharaja forest or from a vegetable field from Nuwara and then use it in a paddy field, it's not going to work. The chances of that biofertilizer becoming unsuccessful is really high. So it is very important to match the ecological requirements of the organism with the characters of the destination. Okay, so it's important to know the ecology, the host specificity characters. Is there a host specificity? So especially with the nitrogen fixers, with symbiotic nitrogen fixers, there's very strict conditions that certain bacteria work with only certain legume crops only. When it comes to mycorrhizal fungi, again, there's host specificity. So we need to have an idea about that and the soil characters at the destination and the characteristics of the targeted agricultural system. Without this knowledge, we cannot do the isolation of organisms to produce a successful biofertilizer. At the same time, at the end of the whole process, we should be able to tell the farmers how much to apply, how to apply the biofertilizer, and how long can they store, under which condition can they store. Okay, so these kind of things should be clearly informed to the farmers. That means to tell these things, there's a lot of research need to be conducted. That means you cannot produce a biofertilizer in 1 or 2 years. To produce a biofertilizer, it takes 2 to 3 years minimum. Minimum of 2 to 3 years will be taken to produce a good quality biofertilizer. Okay, so in addition, there are different types of biofertilizer formulation. Let me brief you about that aspect here as well. So I told you that when we have the inoculum, then we need to convert the inoculum into a fertilizer formulation. So this can be a liquid fertilizer, freeze-dried powder, or a carrier-based formulation. So which type we are going for depends on many factors. So we need to consider the physiological requirements of the organisms, and these formulations help the organisms to hibernate or survive during long storage time. Okay, and quickly activate once we release to the new environment, and also support the establishment in the new environment, maybe provide the seed nutrients and maybe provide protection against the predators once we release it to the new environment. So the biofertilizer formulations can be produced in order to facilitate these requirements. And these the biofertilizers can be monocultures, that means contain a single type of organism, or it can be a mixed culture, that means a mixture of different types of organisms, or it can be a biofilm. Biofilm means like a few organisms cultured together. It's different from the mixed culture, because in the biofilms, the organisms enter into a specific growth condition, forming the biofilms usually become very advantageous when we apply to the field, because it's going to give some extra layer of protection from the predation and facilitate easy establishment. So these are some of the important facts to remember in relation to biofertilizer formulation. Another important thing in this whole process of biofertilizer production is to assess the storage and shelf life. I'm sharing some experience from our own studies and from the Philippines, where they have done a lot of biofertilizer work. One of the biggest challenge, especially when we have liquid type formulations, is to maintain the quality throughout the storage. In this research, you can see how the visual characters change with time. By day 90, you can see visually the material has changed, because the biofertilizers contain live organisms. So they keep on continuing their activities. So because of that reason, when we produce the biofertilizers, the organisms remain active, and especially if it is in the liquid form, the shelf life or the storage life is very short. We cannot keep it more than 3 months, because when the organisms are active, that means there are gases produced, toxic substances produced. Sometimes it may be harmful for the to handle. So the solid formulations and freeze-dried ones are very much favored in that sense, but freeze-drying is very costly. Solid formulations again have challenges, because with time, the viability of the organisms might drop. But in our lab, the biofertilizers we have developed, we were able to maintain the viability at very high level, even up to 8 months into storage. Okay? So this is an example of liquid biofertilizers. If we don't pay attention to the storage conditions and the quality, these type of blastings could happen. This is an example from Sri Lanka with the liquid organic fertilizers that were marketed during the crisis time, which were claimed as biofertilizers, liquid fertilizers, etc. But the important thing is these type of blastings happen because there are live organisms keep on maintaining its activity. Okay, because of that, the gases and toxic products are produced inside and lead to these type of blasting, like harming the human health as well. So,
Thank you very
in
much. Yeah.
summary,
Thank you.
yeah, the biofertilizer production is not an overnight thing. Yeah.
Thank you very much, Warshi. And also an important message is when you work with living organisms, handle with care. That's the important part. Understand psychology, understand the growth rates, patterns, and so on to make sure you make the maximum use of it. When you presented all these things, I mean, it's clear to me that standards play a very important role in biofertilizer. Let us know about, I mean, something about these standards, quality standards, and so on with respect to biofertilizer. Over to you.
Yeah, in Sri Lanka, the standards are being produced. As far as I know, the standard has not been finalized yet, but it is under development. Anyway, one thing we experienced in the massive scale experiment the whole country did in the past 3 years is when there's no regulation on quality control, we run into a lot of problems. Okay, so there can be many fake products coming to the market, uh, like having many resulting in many issues. So when there are no regulations on quality control, it leads to inferior quality products flooding the market and products that mislead the farmers and poor return on investment because we spend a lot of money buying it, but there's no effect of it. And then introduction of invasive species to agricultural fields, which is very important when we talk about biofertilizers, and also health concerns for the users. Because of these reasons, it's important to have standards. So I told you in Sri Lanka, we are in the process of developing standards. I think some members in this STEAM audience might be very well aware about where the standard is at the moment. I know it was open for public comments 2 years back, and it should be now finalizing. So there's a draft standard on biofertilizer specification. And in China, Kenya, and even in the European countries, they have their own standards. In China, these standards are, I think, more than 10 years old because they have been using biofertilizers for quite a long time. So when we look at these different standards, few common considerations can be seen. That is, they all talk about loading of targeted beneficial microorganisms. For example, if the producer claims this is a phosphate solubilizing bacteria-based biofertilizer, then there's a number, the minimum number of bacteria per gram or per milliliter of that biofertilizer that should be there. And then identity of the microorganism should be presented up to species or to strain level. In China, they are requesting to present the identity to strain level, that is more than the species level. Okay, and then the density of the contaminants by its biological and chemical contaminants in the biofertilizer, and the density of pathogens. So, for example, there are maximum allowable limits for E. coli, like coliforms, and also the cadmium, lead, like trace elements. So these are defined in biofertilizers. And also, there's regulations on shelf life. So these considerations can be seen in the standards that are there for biofertilizers. And in Sri Lanka, as I mentioned earlier, the standards are under development.
Yeah, thank you very much, Warshi. I saw a question and a comment on just about the standards quality control in the chat box. Let's come back to that later. One of the objectives of this webinar series is to make sure people rely on facts and also bust myths, especially the misconception. Those are extremely dangerous when you try to propagate those type of ideas without knowing the real scientific background. So why don't you tell us, with your own experience, Warshi, what are the common misconceptions related to biofertilizers that prevails in the country? Over to you.
Yeah. I think by now it is clear about what is a biofertilizer, how is it acting in the environment, and how is it being produced. So with that understanding, let's take a look at some of the misconceptions people have about biofertilizers. I have listed some here. One is like some people think origin of the microorganism in the biofertilizer does not matter. That means some people argue like you can take the best phosphorus solubilizer from the Singharaja forest soil and make a biofertilizer to apply anywhere in the country. I think by now it should be clear to you, it is not so. The origin of the microorganism, from where did we isolate? What is the ecological character of the organism? This matters. Okay. And another misconception is one biofertilizer can be used for any crop. So we have seen this even in Sri Lanka, like some biofertilizers which are marketed, like they even claim like you can use this for any crop, which is not true, because in different cropping fields, the soil conditions, the microclimatic conditions, then the plant microbial interactions, these are going to be different. So the performance and the efficiency of the biofertilizer, we say the efficacy of the biofertilizer will be different from place to place. So one biofertilizer cannot be used for any crop, which is one of the biggest challenge, biggest disadvantage we have for biofertilizer when it comes to when we compare it to chemical fertilizer. Because when you take urea, you can use urea in tea plantations or in paddy fields or in maize cultivated fields. But if you take biofertilizer for these three crops, you need three different types of biofertilizer. You cannot use the same biofertilizer for tea, paddy, and maize cultivation. Okay. And all biostimulants are biofertilizers is another misconception. I mentioned this in my second slide, that at the definition of the biofertilizers, although biofertilizers are a type of biostimulant, not all the biostimulants are biofertilizers. Okay, I hope it is clear to you. Another misconception is compost is a biofertilizer. I will explain to you in the next slide why it is a misconception. Okay, and then fish tonic, like fermented organic fertilizers are biofertilizers, is another misconception. So these are referred to as organic fertilizers rather than biofertilizers. And some people think there are no standards for biofertilizers, which is not true. In many developed countries and also in developing countries, there are standards. In Sri Lanka, we are in the development of standards. Okay. Another misconception is that chemical fertilizers can be replaced 100% by biofertilizers in commercial agriculture. I hope by now it's clear to you. It is not possible if you are talking about commercial agriculture where we plan to take a big harvest and sell it. Okay. So now let me come to the question, why compost or vermicompost is not a biofertilizer? You can consider here the fruit tonic, fish tonic, like the tonics also. Why these are not considered as biofertilizers? One reason is, if you look at the standards that I mentioned related to biofertilizer, in all these standards, it's very clearly mentioned that microorganisms in the product should be clearly identified and satisfy safety and toxicological requirements. But if you take compost, vermicompost, and those fish tonic, fruit tonic, like different organic fertilizers, in these, the microbial diversity is very high, and we don't know exactly who are the organisms living in and whether all organisms are safe. Especially in compost, we know there are coliform type organisms. There can be pathogens depending on the raw material we are using. So we cannot guarantee that it is having organisms that are safe to use, and we know everything about the organism. So that is the principal reason, or the main reason that we can use to say it is not a biofertilizer. And then consistency of the product should not change from batch to batch. As in any fertilizer, when we take a biofertilizer, consistency should not change from one batch to another. But in compost, vermicompost, and those tonics, the composition would change from time to time depending on raw material and the process of composting or the fermenting. And another thing is, should meet the standards on density of beneficial microorganisms with well-identified plant nutrient supplying character. That means if we are saying it has nitrogen fixing ability, we should know how many nitrogen fixers are there, and is it meeting the standards of a nitrogen fixer-based biofertilizer? Okay, so because of that reason, in compost, vermicompost-like materials, we cannot consider as biofertilizers. Another reason. Okay, and also the last one, the free from fecal contaminants and invasive species and pathogens, we cannot guarantee for compost and vermicompost. Because of these reasons, the compost, vermicompost, and the fermented products like the fruit tonic, fish tonic, these are not biofertilizers. Those are liquid and solid organic fertilizers. And why certain misconceptions are there among people? One reason is the mistakes people do in biofertilizer research. So I thought of it is good to talk about some of these mistakes here. One biggest mistake is not evaluating ecological traits of the microorganisms used and not declaring about these ecological requirements of the organism that they have in the product. Okay, this lead to wrong use of the biofertilizer, very ineffective results. And also not using proper controls to assess nutrient responsiveness of the crop in a given soil. This is a very common mistake I have seen because I have been involved in many evaluation of many MPhil, PhD theses and research articles written related to biofertilizer research in the country. What I have noted is like in many instances, the people do not use proper controls. That means we do not know whether there is a fertilizer responsiveness in the soil to begin with before they are starting applying the biofertilizers. Okay, so it's very important to understand, is the soil showing fertilizer responsiveness or the nutrient responsiveness before we starting this when we are doing this biofertilizer research. And also in most of the cases, the research use farmer practice of fertilizer application to compare the effectiveness of the biofertilizer. But if you take farmer practice, it is not a standard. From farmer to farmer, the practice would change. From region to region, it would change. So using farmer practice in the comparison is not a good thing in scientific research. They should use a standard positive control when they are doing the biofertilizer research. And not reporting crop variety and not collecting important data and parameters is another mistake in biofertilizer research, and not having enough replications in time-wise and with space is another issue, and not investing enough time to gather required details during the development. For example, not looking at the toxicology or the health concerns or characterizing the growth rates, etc., of the organisms. So this is another issue that we see in biofertilizer research in the country. And also, most of the people stop the research work until inoculum production and do not proceed into formulation of a biofertilizer. So having an inoculum, developing an inoculum is not developing a biofertilizer. So there are additional steps required to complete it, the inoculum into a biofertilizer. So these are some of the common mistakes that we see in biofertilizer research in the country. And the biggest issue that we see is generalizing the results, that maybe they have done a research in a pot experiment scale or in a single location, but then generalize the results to the whole country or all the context, all the crops. This is one of the biggest issues that we see in relation to biofertilizer research. And also not identifying the microorganisms up to at least to species level, and not analyzing their toxicological characters is another issue that we observe in the biofertilizer research. So if you look at this whole thing, I think you might feel a bit negative, right? In relation to biofertilizer, there are a lot of problems. But even though there are problems, there are solutions. Okay, so to clear the misconceptions, we need to work in a very scientific and strategic way with biofertilizers, and there are solutions for every problem. For example, if there's a problem of introduction of invasive microorganisms, inefficient products, Then we need to work on selection of suitable competitive strains for specific climate regions, crops, and soils, which now can be done even using with the assistance of molecular techniques. Okay, then and rather than just depending on the traditional culture-based techniques, there are molecular-based techniques helping us to isolate, identify very effective strains. And then if there's a problem with low quality products and lack of vital cell viability, and another big issue that which I did not highlight earlier is mutation of microbial cells during multiplication in fermentation, because we are talking about live organisms. These organisms keep on evolving. So during our storage, during our processing, these organisms keep on evolving. So therefore, it's important to have major quality assurance steps with time to make sure the product you are developing has the intended quality. And another problem, especially lack of interest in the private sector on biofertilizers, is the high investment cost. So therefore, there should be government interventions and then good collaboration with the industry and the academic institutions to research on alternative growth media using industrial byproducts, which can bring down the cost. Okay. And another issue is poor understanding of importance of microbes for the below ground processes. So creating the farmers' awareness about these biofertilizers and the importance of soil organisms, improving the soil ecology, will create interest among people to use these products instead of only chemical fertilizers or organic fertilizers. So creating awareness is also very important to popularize biofertilizers. I hope I have answered your questions, sir.
Thank you very much. Thank you very, very much. It was a marathon session, I must tell you, Warshi. Giving a lot of information, and session does not end because there's a large number of questions. I even had to stop receiving questions because of the interest of the time. So Warshi, thank you once again. And I'm going to the Q&A session right now very quickly. It's unfair to ask from you to be short and crisp, but I have to do that because of the time. It's late right now because we have to keep this audience go and have their dinner and have a good night's sleep as well, not having a lot of problems in their mind. Some of the questions you have already answered. So I will read the question and skip them rather than wasting our time or trying to repeat the same thing over and over again. The first question is posted by Deeptika Kakulandara. The question is that what type of biostimulants or biofertilizers are tested and recommended to be used in Sri Lanka? Over to you, Warshi.
Yes, as far as I know, the biofertilizer, I told you all biostimulants are not biofertilizers. There are biostimulants which does not have any live organisms, which have been tested and recommended to use in Sri Lanka, which contain only plant growth hormones like products. But if you look at the biofertilizers, according to my knowledge, only the biofilm biofertilizer produced by NIFS, the National Institute of Fundamental Studies, has been tested, and to some, I don't know whether it's a recommended, but it's approved to use in the country. Marketing has been approved for this particular biofertilizer. There are several other biofertilizers which are being tested under technical evaluation program of the Department of Agriculture. So if you ask me like how many products are there that have been tested and recommended to use, I don't know whether it is recommended, but approved to use is only one product in the country.
Yeah, thank you very much, Warshi. The second question you have already answered, posted by Rasika, saying that why can't we consider microbial products or metabolites as biofertilizers? So you have done it, respond to that similar question several times. The next question asked by Nuwan goes to the history actually. How did the farmers grow crops before the invention of chemical fertilizers in 1923 in commercial scale?
Mm-hmm. Yeah, so there have been we were catering to large populations in the history, in the past, and people used a lot of organic fertilizers, not in the sense of fertilizers, but they have applied organic materials into the soil, and there were a lot of regenerative agricultural practices that farmers used at that time. And by 1920s, I think in certain countries, chemical fertilizers have been already introduced, but in Sri Lanka, as far as I know, we were using only guano-like fertilizers, which are not chemical fertilizers. But there were a lot of organic methods that people have used. But the term biofertilizers that we are using is a very standard, specific material. So such a material was not introduced until 1950s. The first introduction, as you may remember from my presentation, was in Soviet Union and China. In 1950s, they used these organisms. But before that, of course, farmers used different other nutrient sources. At the same time, the cultivation intensity is not as high as today. Even though there were commercial cultivation, the intensity of practices and the number of crops per year is not similar. And there were a lot of rotations that has been practiced. At present, we are not going for such fallow rotations, which give enough time for the land to recover. I hope I answered the question.
Yeah, a lot of other factors coming, but you are quite correct. The population, the land available to do those things, all those matters a lot. Thank you very much. Guru Singh asked this question: can we fulfill the total plant nutrient requirement from biofertilizer? You actually responded to that earlier, so I'm not going to take time on that. Then Kusal Herath asks this question from you: use of biofertilizer, not stimulants, on 100% organic lands. I give a lot of explanation on that. Now, will those be considered as organic, or any possibilities in detection of chemicals in the end product when you use 100% biofertilizers in organic systems?
I think a very good question, because when we talk about organic cultivation, especially if you are considering about certified organic crop production, there are certain requirements to be met to claim that we are producing it in an organic manner. So in organic crop production systems, there are authorized materials that we can use. Even a biofertilizer, as you can see in the production process, it can produce using synthetic materials as well. I mean, like, organism is live, of course, but the carrier material and the growth media can contain synthetic material, or we can use natural materials as well. So biofertilizers can be produced targeting organic crop production. So I emphasize it here. If you are planning to use it in organic crop production for certified organic farming, the biofertilizers should be produced targeting that production system using the authorized materials. And with that, biofertilizers can be used in organic crop production. But again, I think there was another part of that question which asked about can we fulfill the nutrient requirement? Again, here it's important to understand the cropping system and the intensity of the removal of harvest that we are expecting. So if we are expecting to remove harvest very frequently in large quantities, biofertilizers alone will not be enough, even in an organic system. There are enough research, meta-analysis, considering thousands of research done in Europe and in China, that indicate in organic systems, when biofertilizers were used in organic systems, phosphorus and potassium mining from soil has been found as a frequent problem. So if we are assuring that nutrient mining is not happening, that means we are supplying the biofertilizers together with organic fertilizers approved for an organic farming system. Of course, biofertilizers can be very effectively used in organic farming as well.
Thank you very much, Warshi. Parakram Vijayasinghe made a comment as well as then question emanating out of that. The comment is that the proposed standard for biofertilizer includes a very important condition. That is, they shall not pose a threat to humans or other organisms. This is the proposed standard. And the question that comes out is that shouldn't this condition be imposed strictly for chemical fertilizers, which are well known to be harmful to all microorganisms in the soil and to humans? Are we turning a blind eye to this problem in chasing after chemical fertilizers as the preferred option? What's your opinion, Warshi?
Okay, so let me enter into that question this way. So if we look at the two types of fertilizers, the chemical fertilizers and the biofertilizers and the organic fertilizers, all three. So there are contaminants in that can be there in these fertilizers that could be harmful if we are using it at a certain concentration, or if the material get accumulated at a certain concentration that the chemical fertilizer or the organic fertilizer can create health hazards. So that is, let me reiterate, it relates to how you use it. If you are using it not following recommended dosages, of course, and also if you have imported material which does not meet our quality standards, which have been developed considering these health hazards conditions, then of course these materials can create problems. However, when it comes to these biofertilizers and some organic fertilizers which contain live microorganisms, the problem is the harm does not relate to the concentration but to the organism itself. The organism, whether it is a single organism or present as 100 or 1,000 or 10,000 in the material, it might be secreting substances which might be skin irritants or cause diarrhea or some other problem. Very clear problems that are linked to the organism itself. So irrespective of the concentration. So that's where the problem lies. Like with the chemical fertilizers and some organic fertilizers, we can control the hazardous nature by regulating the quantity that we apply. But when it comes to biofertilizer, it is difficult to regulate this hazardous condition by regulating the quantity that we apply. If it is even a single member, the problem might be there, because once you apply to the environment, in the environment, the organism can proliferate, multiply, because we are talking about live organisms. And that is why the health hazards related conditions are very strict on biofertilizers, but not on chemical fertilizers. But I agree with you, like if we misuse the chemical fertilizers, not only chemical fertilizers, even organic fertilizers, if we misuse in terms of the using in the farms as well as at the time of releasing to the market, maybe importing or locally produced, not meeting the quality standards, then the material can be problematic and hazardous, and we should have strict regulations imposed on those things as well.
Thank you very much, Warshi. Damiru Setuge asked this question: how can we use biofertilizer for improving the efficiency of ERP, epoxy rock phosphate? Are there any recommended product or method for pre-application formulas with ERP?
I know several research groups working on phosphate solubilizing microorganisms that we can apply with ERP and also apply to ERP to increase the solubility. So there are some, I think there's a group in Rajarata University who is trying to develop a microbial inocula that we can apply to ERP to increase the phosphate soluble phosphorus concentration in the material and then release it to the soil. So we also tried it with the biofertilizers we have developed with ERP. But again, in some places it worked, but in some other systems it did not work. So there was a lot of inconsistency in solubilizing phosphorus from ERP according to our results. But this is an area that is being researched.
Thank you very much. Bandar asked this question: can we use biofertilizers for certified organic cultivation without using other manures?
I think I answered this question earlier as well.
So, yeah.
So the organic, so for certified organic cultivation, you need to have biofertilizers produced using the ingredients that will not harm your certification. And again, depending on the ecological state of the farm, I mean, like if you have a very well balanced, fertile soil developed in terms of biological properties, very well improved soil developed through the organic cultivation system, applying biofertilizers will be helpful. But as I mentioned, previous research indicate long-term research done in many places in the world, analyzed together, indicate that in organic systems, using biofertilizers alone will lead to nutrient mining from soil. So the farmers need to be careful. Especially this happen with respect to phosphorus and potassium nutrients, not with nitrogen. Okay?
Thank you very much, Warshi. Prashanti asked two questions, so let me pose these two questions together. Isn't the chemical fertilizers harmful for biofertilizers? Then if so, why incorporate them together? That's the first question. Chemical fertilizers and biofertilizers. And then the second question is, can we use dry biomass as biofertilizer? Would you?
Yeah, so I think a very good question. So chemical fertilizers and biofertilizers can be used together. That's what we have shown in our research, because with the phosphate solubilizing bacteria, we applied it with 30% reduced rate of TSP. So that means together with the biofertilizer and the TSP we have used successfully. And the cyanobacteria-based biofertilizer developed by Dr. Vajira Balasurya, she also used it with 25% less urea application. So the important part is you need to isolate the correct organism. That means you need to isolate organisms who would perform the intended act, even though there are some amount of available nutrients in the environment. Okay, so that's why I said, like, if you isolate a phosphate solubilizer from Singharaja forest, where the phosphorus availability is very, very low in the soil because of that close nature of that nutrient cycling system. So that organism has evolved in a situation where where it would perform its activity when available nutrient levels are very low. If you bring that organism and apply it in our agricultural soils and apply it together with a fertilizer, it will not work. If you want to have an organ like a biofertilizer that would perform well in our agricultural soils together with fertilizer application, then we need to isolate organisms who would perform their task in presence of certain level of available nutrients, or would not get inhibited by the chemical fertilizer application. Now you might ask, why do we have to apply chemical fertilizers if we are using biofertilizer? One reason is, according to research, what we have observed is sometimes you need to give a kick. In simple terms, if you say, you need to give a kick to kickstart the growth of the plant, okay? Because when you apply the organic fertilizers, biofertilizers, sometimes you cannot give the nutrients at the rate that the plant requires at the very first instance. So you need to have some kickstart with some level of inorganic fertilizers supplying readily available nutrients in the soil solution. That's why we have to, we are using the two things together. Okay, applying biofertilizer alone may not work in most of the commercial fields, because the organism will not be able to supply the nutrient at the rate that the plant requires. Okay, so that's why we have to supply the two things together. So you need to isolate the correct organism.
Yeah. Thank you very much, Warshi. I mean, I can take only two more questions. There are a lot more, Warshi, but in the interest of the time, I will take only two. These are last two. First out of the two, from Deeptika Kakulandara. Similar question with respect to ERP that you responded, but let's see whether there's a difference here. Is there a possibility to produce a biofertilizer introducing P-solubilizing microorganisms together with ERP for low phosphorus soils?
This is again being researched. One part of research done by, I mentioned about Ms. Kumudu Kumari Navaratna from HODI. She's working on vegetable crops, trying to see whether we can use ERP to partially replace the TSP requirement if we are using it together with a biofertilizer, PSB biofertilizer. And also, I must say that the ERP has been successfully used in wetland paddy cultivation, not in the wet zone paddy cultivation, where we have very acidic soils. In acid sulfate soils in Nilwala Basin and in Bentota-like areas, in these places the pH is low, so the solubility is high. So in these places, ERP is being used effectively. But this I cannot tell exact answer because this is being researched, still under investigation, according to the experience of our lab as well. So in some places it works, in some places there's still a lot of inconsistencies, mainly because I think we did not find the correct organism to work in the system. So this is an area that needs more research.
Yeah, thank you very much. This is the last question. I'm sorry once again, colleagues. The time does not permit me to move further. The last question is from Mahesh Lianage. Last question that I'm posing to you, Warshi. As biofertilizers are very specific and their performance varies spatially as well as temporally, then how a layman farmer can use biofertilizer products available in the market effectively?
Very good question. I would
Good question to end, isn't it? Yeah.
Good question to end the session, I think. Yes, this is all depend on how talented the research group is, I would say, because as you can see from this whole presentation, there's a lot of problems with biofertilizers. But if you have the correct combination of organisms, if you do your research well and identify the correct group of organisms to include in your biofertilizer, you can face this challenge. I can say it confidently because we did so in our research. So, of course, we cannot produce a biofertilizer similar to urea or to TSP that would work in any crop, any cropping system. But at least we can try to have develop a biofertilizer which we can recommend for a large area for a particular system with a particular character. For example, we have developed a phosphate soluble acid biofertilizer in our lab with the support of industry and also the Department of Agriculture. So this one we can confidently recommend in dry zone and the intermediate zone, not telling like, okay, this is only for farmers in Anuradhapura or this is only for farmers in Polonnaruwa. No. So we have developed a biofertilizer which worked very well when we did field trials in dry zone and in the intermediate zone without a problem. So that all depends on the skills of the research group, I would say, and how much thorough work that we do at the beginning in the isolation step to isolate the correct group. And I must say, if your biofertilizer contain only a single organism, because there are many biofertilizers containing single organism or just 2 or 3 members, such a biofertilizer may not be very well adaptive to different conditions. It will be very specific. But if you have a biofertilizer which contain a consortium that include like 5, 6 members or 10 members, then such a biofertilizer has high chance to perform well in a range of environmental conditions.
Thank you very, very much. Yeah, Professor Warshi Dandeniya for being with us today and accepting our invitation to deliver this very informative presentation. I'm really sure and positive that all the participants got a good knowledge about the factual part of biofertilizers and got cleared about the myths if they had in their mind and clarity of the scientific matter. So, colleagues, thank you very much once again for being with us, having confidence on us about the webinar series that we have conducted. This is for the 12th consecutive time. We want to meet you again in 2 weeks' time on a Wednesday, starting at 7 p.m. and coming to your places to solve most of the mysteries that you have had in mind, especially in terms of scientific aspects. So thank you very much once again. Have a good evening and good night. Thank you.
Thank you, everyone.